Regulator Effects creates hypotheses for how downstream diseases and functions may be driven by the predicted activity of upstream regulators, in effect explaining the impact of upstream molecules on downstream biology. This may help provide potential mechanisms for a phenotype or drug, define drug targets, or help you discover novel or confirm known regulator to disease or phenotype or function relationships. The algorithm works by finding statistically significant and causally consistent intersections between upstream and downstream regulatory networks as shown in this diagram. IPA then annotates the constructed networks with known relationships between the upstream regulator and the downstream disease or function if they're known. A new Regulator Effects tab in IPA shows the results. This tab is automatically populated when you create a new analysis in IPA and displays a lot of information about each regulator effect network. I'm going to hide the columns that display counts to make more room. This analysis is of cardiomyocytes that have been differentiated in vitro from embryonic stem cells. The networks are ranked by consistency score, which is higher for causally consistent and densely connected networks. The details of the algorithm are explained if you click the More Info link at the top right. For each network, the Regulators column lists the upstream regulators, color-coded where orange indicates a predicted increase in activity and blue a predicted decrease. The Targets column lists the relevant targets from the dataset, and the Diseases and Functions column displays which of these are in each network. The last column annotates the networks for whether there are known connections between the regulators and the diseases or functions. Many of these columns are filterable, so you can focus on the networks of interest. Here I'll filter on functions relevant to contractility. Clicking the link at the left opens the Regulator Effect network. The top layer contains the predicted regulators, the middle layer is the relevant dataset targets, and the bottom layer is the diseases and functions. Orange and blue lines indicate causal consistency, whereas yellow lines show inconsistency. Another way to add information to this visualization is to show each item in its predominant subcellular location. Clicking this button creates a sort of instant pathway where you can see cases where extracellular factors are likely activated to drive signaling into the nucleus to activate or inhibit transcription factors. These lead to effects on diseases and functions shown at the right. You can also regenerate regulator effects networks to answer different research questions. For example, you can limit the upstream regulators to transcription factors or find compounds that may increase or decrease downstream effects. You may customize these networks however you wish. Regulator Effects is available in IPA now and is automatically included in your IPA license.